Today I am talking about RNA isolation. But the question is why? Why we need RNA isolation? The answer is, for example, we have wild type animal and mutant animal, and we have a question that in these mutant animals, some of the gene in the cardiomyocytes, for example, TTN, MYH7 or FOS gene, they are upregulated. Or for example, we have a hypothesis that in the odontoblast of this mutant animal, the DSPP, DMP or OC genes, they are downregulated. Or we have a hypothesis that in these mutant animals, the Purkinje gene in the neuron is upregulated. So when we are really looking about the expression of some genes, either they are downregulated or upregulated as compared to a control group, we need RNA. Or, for example, we have designed a drug and we have treated these cells with that drug A, and we have treated these cells with a placebo control or with water or DMSO. And our hypothesis is that the administration or treatment of these drugs in these type of cells increase the expression of some genes or decrease the expression of some other genes. So when we are really talking about the expression or messenger RNA transcription of some of the genes, then we need purified RNA. How we isolate RNA? I will walk you through about the isolation of RNA from the osteoblast. This is a mouse femur. I have harvested it. First of all, I will flush out the marrow because I don't need marrow. It has stromal cells, it has hematopoietic stem cells, it has TB cells, it has leukocytes. I don't need them. For example, I don't need them. I just don't need them. So I will flush out the bone marrow. I will cut these two parts because these parts have chondrocytes, and I will isolate the diaphysis where I will get maximum number of osteoblasts, and I will crush the bone and I will add trizol. What trizol do is it actually lyses the cells and exposes the cytoplasm, which is the area where there is RNA. So I will put the crushed bone here and I will add trizol. I will add 3.2 millimeter stainless steel beads and put this tube in the bullet blender and adjust the bullet blender at 8 speed with at 4 degrees centigrade and for 5 minutes, so that the bullet blender physically crush the bones plus the trizol will lyse the cells. After that, I will take out the tube and I will put 200 microliter, not milliliter, 200 microliter chloroform, and I will vortex it. The chloroform is known for the phase separation. It will be helpful in the separation of different phases of organic molecules. I will put it on ice for 15 minutes as RNA is not thermostable. That is why I will try to keep the temperature as low as possible. And then I will centrifuge this tube at 4 degrees centigrade and the speed will be 12,000 RPM for 15 minutes. And after 15 minutes, I will see three phases. The upper phase, which is quite transparent, the bottom phase, which is trizol plus organic chemicals, and an interphase, which is a cloudy layer. The RNA will be at the top layer. This layer, the transparent red layer, has RNA. The interphase and the bottom layer don't have RNA. If this phase will enter in the supernatant, it will contaminate the RNA. So I will take out the supernatant. And after that, I will put 500 microliter isopropanol. Isopropanol is known for precipitation of RNA, and I will put this tube in minus 20 degrees centigrade refrigerator or freezer actually for 1 day. I can put it for 1 hours even, but I have optimized it in a way that 1 day is working for me. With 1 day, I am getting a more purified and good quantity of RNA. And then after that, I will take the tube out and I will centrifuge the tube at 40 centigrade for 45 minutes, and the speed will be 12,000 RPM. After that, I will remove the supernatant. Supernatant will go out. Now my RNA is in the pellet form. I will wash the pellet with the 75% ethyl alcohol 2 times, and ethyl alcohol will remove the debris, some of the isopropanol and some other contaminant which are there. It will remove it. I will air dry my RNA. Now I have a very purified RNA at the bottom of this tube, and I will dilute it with 20 microliter of water, and I will go for quantification and for checking the quality of RNA. Then after that, I will make cDNA and will see that the genes which I had included in my study, and I had a hypothesis that whether these genes in the mutant Y or wild type in the mutant animals are upregulated or downregulated as compared to the wild type. For that, I will do qPCR. How I will quantify RNA, how I will make cDNA, and how will I do a quantitative PCR. This topic I will cover in the next video.